I need to get my homework.
And then just like disappears. Oh. Is she calm?
Okay. Sorry.
No, you're good.
Good.
Okay. So does somebody wanna start it?
Yep.
Okay.
I'll read the first question. You good?
Mm-hmm.
Should I read the first question?
Sure.
Okay. Elk have 68 total chromosomes. How many total chromosomes would be found in the following types of elk? A tetraploid elk. For A. And for a tetraploid elk, it's 136, which comes from, wait, no. Is that the answer? 136?
That's right.
Let me make sure.
For A,
Yeah.
Oh.
what the heck did I do?
Is that what, huh?
So A is 136?
Mm-hmm.
Yeah. I'm not sure.
It is.
Dang it.
Oh.
So did you just do 4 times 68?
No, because 4 times 68 won't come to 136.
Yeah, I know.
Yeah, I originally did 4 times 68, and then I looked at the answer key, and then I was like, crap.
I
So
wrote a
why?
note in here that, well, I don't know if it's right. I wrote a note in my notebook. I'd like put a question mark on it. So I'm not sure if that's the answer.
See, on B, I got 136, and that's like 68 times 2. But
Yeah.
on A, I got 204, 68 times 3, because I thought it was a triploid. So you're supposed to multiply 3.
On A, I said a tetraploid is 4N. I thought. This is my thought process, and so I did 4 times 68 and got 272.
Okay, but an elk is normally diploid. That's 2N, and 2N equals 68. Because normally we're all diploid.
Mm-hmm.
So 2N, and it normally has 68 chromosomes, so 2N equals 68.
And then just N would be 34 then, and then 34 times the 4 would be that then.
Yeah.
Oh.
Okay, crap.
Yeah, write that down.
No, because I did exactly what Lindsay did, and so when I looked at the key, I was like, what the F? And then now I get it.
Yeah.
Let me change that. That makes a lot more sense.
Yep.
I should probably start checking the answer key.
Yeah. I always do that like every time as I like work my way through them because otherwise I just get too confused. It like helps me to have something to reference and like look at and make sure I'm doing it right.
Yeah.
So A is actually 136.
Yeah.
And so B would be 2 times 34, which is 68.
For a diploid?
Yeah.
Yeah. Because it's just disomic.
No. It's disomic for chromosome 5, which means. Hold on.
Wait, do we know if the key right now is correct?
Oh, yeah, if it's disomic for chromosome 5, then that's right. Yeah, the key's got to be right.
Okay, then yeah. I have the key up, and so it says it's 68.
Okay, so A is 136, B is 68, C would be...
I think I'm going to need to redo my first one. I don't think I understood it properly.
Yeah.
Yeah, C would be 69 because if you're trisomic for one chromosome, you have the normal amount of chromosomes for everything else, but then you have one extra on chromosome 17, so you just add one.
Okay, and then D would be 67 then.
Uh-huh.
Okay, that makes sense. So it says, in the question, it says that elk are diploid with chromosome number of 68. So we know that N has to be half of that.
Mm-hmm.
Okay.
Yeah.
All right. I feel better about that.
Yeah, because like all organisms are like all like animals usually. I don't know actually about all, but like usually are diploid, and then the gametes are haploid. So, anyways,
Sweet.
okay. And then wait, so E, an elk that has suffered a reciprocal translocation between chromosome 5 and 17, just switched, but
I said it was 34 still.
why is it 34? It's a diploid organism.
Yeah, but the question, so it says, let me read it again. Is that, what does the key say? Because that could be,
Six,
So it says 68.
okay, not that it's a reciprocal translocation between chromosomes 5 and chromosome 17. I said 68 originally, I just reduced it because I figured it was wrong.
no, it's 68. Because 68 is the normal amount of chromosomes it has.
Okay.
Yep.
Because the reciprocal translocation doesn't alter the amount of chromosomes.
Yeah, you're just switching them.
Okay.
So, yeah.
Mm-hmm.
Makes sense.
Okay. And then question 2 says, this question concerns the human diseases that we discussed in lesson 7. Below is a list of terms that we've learned about recently. So polyploid, triploid, paracentric inversion, deletion, monosomy, duplication, aneuploidy, reciprocal translocation, trisomy, and non-disjunction. So well, it goes through all the different syndromes. So how do you say the Patau syndrome?
Yeah, and you just go through the. Did you guys just use the table from the
Well,
Wait, what table? Oh,
the table in the PowerPoint?
no, I didn't. I like
That's what
was I
maybe
just
I got it online. Let me make sure that I'm not making things up.
Wait, I didn't know there was a table. What slide number is that?
Yeah, there's a table, but no, this is a different one. I looked at a table that I found online, but the table that's on that is on the PowerPoint is on slide 4, but so it talks about the different syndromes.
Okay.
Oh, okay.
But I used a different table. I used a different table from online.
Okay.
So for A, is it aneuploidy and trisomy, or how do you say that?
Mm-hmm.
And polyploidy and non-disjunction.
Oh, all 4 of those?
Oh,
Yeah.
Yeah, I thought it was just
so
name one.
on the
And it's
announcement, she
which
said
one.
that Key was wrong in which none of them are polyploid.
Oh, really?
Yeah, because polyploid would imply that like each set was like the whole entire set was copied, but none of them are like that. And so she said that the polyploid part was wrong, but everything else is alright.
Oh, okay.
So it's
Oh.
3, it's aneuploidy, trisomy, and what is the third one?
And non-disjunction.
Non-disjunction.
Okay, let me add that.
I only got the 2. I didn't get the non-disjunction.
Yeah, I didn't either. So B, I got aneuploidy, trisomy, non-disjunction.
Yeah.
Okay.
Yeah.
And then C should be monosomy, aneuploidy, and non-disjunction.
Yeah.
I'm missing all these non-disjunctions ones.
I am too.
So the first 3 are the only 3.
But I guess that would make sense because non-disjunction is where it doesn't separate correctly. So aneuploidy and non-disjunction have to go together, right?
Have to go together.
I think so. I need to look at my instructor notes again because I'm.
On the answer key, for the most part, the aneuploidy and the non-disjunction go together, so yeah. Ow.
How would you guys define aneuploidy?
I defined it as changes in the number of chromosomes within a set.
Okay. And
And
that's
so,
due to non-disjunction.
yeah, non-disjunction I defined as like a defect in chromosome segregation. And so, when it can't separate like correctly, then there would have to be a change in the set because you would have either too much or too little. And so that's what I thought of it as. But I totally forgot that that vocab was there, and I did not put any on that except for B. So I missed C and A on the non-disjunction part.
That makes sense though, because if I don't know, they would go together.
Yeah.
Yeah, the slide says that, well, non-disjunction would produce aneuploid cells.
Yeah.
Okay.
That's for meiotic non-disjunction.
Am I reading the right thing?
I guess it can happen during. Okay, so it says it can happen during mitosis, the same thing,
Mm-hmm.
but for somatic cells, it's depending on the cell, it's not going to make as much of a difference. If it's like one somatic cell versus like a gamete. Anyways, okay. Okay, so for D,
I just got deletion.
Yeah, the answer is just deletion because it says polyploid and deletion, but it's not polyploid, so it's just deletion. So we're good.
What is polyploid? Is just having over 3.
It's being not diploid or haploid. It's like more than that. Like you have more than 2 sets of chromosomes.
Yeah.
So a triploid would be considered a polyploid.
Polyploid.
Yes. Because that would be 3 whole sets.
Okay.
Yeah.
And what did you guys get for E?
I originally wrote duplication, but that's wrong.
It's reciprocal translocation. I think it says it in the instructor notes too.
So it's just the translocation, would aneuploidy trisomy be part of it too?
I don't think so, because you're not changing the number of chromosomes, you're just changing the like order of things.
Okay.
Yeah. The key just says reciprocal translocation, and that's it.
Yeah. Let me find it really fast. Yeah, it just says in the instructor notes it's caused by a translocation. It doesn't say anything about anything else. It's a different type of Down syndrome.
Hold on a second. Does the key just say research translocation?
Uh-huh.
Okay. Alrighty.
So, number 3. Okay. So, I have to find my notes on this.
I don't know if I got this right. I should probably pull up the key.
Did you do the video? Did you follow the video?
Did she make a video?
Yeah, she made a video like Tuesday or Wednesday night.
It was Wednesday night.
Oh.
Yeah,
It was later.
it was later. And then the moment I submitted it, I looked at the announcements and I was like, Flip, are you serious?
Well, I didn't do mine until later because I had so much other homework for other classes, and so I like didn't even get my thing turned in until like 12:30. But by the time she had already put the video up, and I was like, oh well, guess I'm using this to do my homework because I need all the help I can get. So
I should look at that.
it makes a lot of
What did
sense.
you guys get for?
Yeah, the video made
I
a
kind of looked at the PowerPoint is where I referenced it off.
Let me pull up the lesson key. Let me find my assignment for this because I don't remember where I put it. Somewhere in here.
The only thing I got frustrated with was that like on the key it said like questions about it, but then on the actual group problem like worksheet it was just like note this, and I was like note what?
Oh, yeah.
And so when I looked at the key and realized that there was actual answer things to it, I was like, what? Okay, I give up on you.
Um, so, okay, we'll go through the key, and my key matches my homework because I did it just like the video said to do it. So you have, let's see, hold on. Let me just read the problem really fast. So suppose an individual is an inversion heterozygote. The order of the alleles on the homologous chromosomes in question is shown below. The little dot represents a centromere. Note that the paternal chromosome has a normal allele arrangement. The maternal chromosome contains the inversion. The capital letters represent dominant alleles and the lowercase letters represent recessive alleles. Suppose that these two chromosomes replicate prior to meiosis. Note that now the paternal chromosome consists of two sister chromosomes or chromatids, and the maternal chromosome consists of two sister chromatids. So when you do that part, so if they replicate, the paternal one is normal. So you have like A, B, C, D, E, F, G, H, I, J, centromere, K, L, M, N, O, and then A, B, the same thing again underneath it because it's the same chromosome. It's like just replicated. And then when you do the mom's chromosome, so she like has, so since it's switched at the E and F, you like flip it and then work backwards. So you get this like weird looking.
I need to pull up the key.
I'm just gonna hold this up, and if you guys want to look at it, cool. It's this whole. Wait, can you even see that?
Yeah.
Yeah.
Flip it where the E and the F are, and then you have like you write the letters like going backwards. So you like they stay in the same order, but you're like, but they don't. I don't know how to describe it. It's super hard to describe. I need to pull up my original because I didn't write the original thing on here. Yeah, so you like flip the F and the E so that they line up to the paternal ones. And once you flip them, so the E is going to have, coming off of that, D, C, I, J, centromere, K, L, M, N, O, going backwards, like off the, like to your left. And then on the right hand side of it, it's going to be the F, G, H, B, A. So you just flip them so that they match the paternal chromosome so that they can actually cross over. And you're going to get basically, so two normal paternal chromosomes, two inverted ones that are that weird formula. And then one of the paternal chromosomes doesn't get crossed over, and one of the maternal ones doesn't get crossed over, so they just stay like that. And then the two in the middle get crossed over, and you have this weird thing with like two centromeres and like just a really weird formula.
See, I was hoping like one of the YouTube videos would show more of what was going on, but I didn't really see. I mean, did you guys see any YouTube videos that really explain translocation or inversion?
No.
Yeah, neither.
Like, it makes sense, but like it makes sense if you like read the notes, and then it makes sense if you like watch her video.
I need to watch her video.
Yeah. But anyways, just know that those, like the weird formula, it's on the lesson key, and it makes more sense when you do it that way. And then basically you get two, so you get four, you end up with 4 gametes, and 2 of them are viable. One of them is a normal gamete, the parental one. One of them is the maternal one that has an inversion, but it's still like a viable offspring because it still only has like, it just has 1 centromere. It just has a weird inversion, but it could still be a viable offspring. And then you get two non-viable offspring. So one is this dicentric chromosome, so it has two centromeres, and the one doesn't have any centromeres. So
It wouldn't be viable.
yeah, so neither of those two, the ones involved in the crossover, are not viable. Like, they'll still develop, but they can't, that individual can't reproduce.
Yeah.
Yeah, it said that like the one that was like the one without a centromere, it said that it would be lost during meiosis and it would probably die, and then the other one, because of its two centromeres, when it goes to anaphase, it's going to be torn apart and then basically not used at all. The only thing I didn't get from her video was like, you know how you do like the J I C D E and then F G H B A. The only part I didn't get was like from the F to the H I understood, but why is the end also switched? Like why wasn't it like A B C D E F G H I J instead of like? Did that make sense? I don't know what I'm
What are
saying.
you talking about?
You have to stay in order. You like keep the original chromosome is that way, and you have to keep it in that order. Like, you can flip the E and the F because that's where they're gonna cross over, but like you have to keep them in the same order that they were already in. Like,
Yeah, but that was my question. Are we copying from the parent, or are we copying it from one of the normal and the crossover?
what?
Oh, never mind. Forget it. Never mind.
I just don't know. I just don't understand what you're asking.
Yeah, I think I confused myself. Never mind. Because when I watched it, I understood, and now I don't understand. But forget it. Oh, it'll be okay.
No, it's still like kind of confusing, but yeah, I don't know. I just need to go back through my notes some more and through like the videos and stuff, but I feel like I don't know. Like, I get how to do it. So, I don't know. Okay, I know that's kind of confusing, but I mean, basically, you end up with two viable and two non-viable offspring.
Okay.
And the ones involved in the crossover, one of them's going to be acentric and one's going to be dicentric.
That makes sense.
Yeah. So, that's number 3. Okay, so let's do number 4.
I'm going to put a star by number 3 so I go back and figure that out.
Yeah, I'm pretty sure this is gonna come a few times on the exam.
Probably.
Yeah. I'm not sure if I did number 4 right, but is that what you guys did?
Let me read through it.
I kind of gave up on 4.
Oh, I gave up on 4 too.
Hold on. I don't think I even got this far. Let's be real, it was 12:30 at night. That was a late night.
I guess I could take the key. Oh, I only got on number 3. I only saw that there was one part to it, so that's why I got a 90% on my homework.
Oh. Well, in the group problems, there was.
Yeah, there was one question for number 3. I didn't see all these, which of the 4 chromosomes are expected to produce viable offspring or anything like that until I saw the answer key.
Yeah. Yeah. Because it just says the arrangement of alleles. Yeah, it doesn't say to do anything else. I just followed the video and just wrote down I took notes basically on her video, and when I did that, I just worked out the problem at the same time and then took notes at the same time and then uploaded that, and I guess that was right.
I should do that because seeing her work through it step by step really helped me on that one. I don't remember what
Yeah,
unit
that
it
one
was.
she did. Yeah, I know. Yeah.
Gene mapping.
Yeah,
Yeah.
Yeah, that one. That
Oh yeah,
one.
which how'd you guys do on the test?
I made like a 75.
76.
That's not like terrible. I mean, hey, passing,
Get degrees, I guess.
Definitely better than the last one.
Yeah.
Oh yeah.
You what?
So definitely better than the last one.
Oh yeah. I did better than my last one too.
I feel like the last one everyone failed.
Man. I'm just like missing some assignments from like the beginning of the semester. Like I've done them all like since like week 3 or whatever, but I'm just like missing a few things. So like my test grades have been helping raise my grade. It's been good.
I think it's mostly the homework that's helping me.
Yeah.
I just got frustrated because I did well on my test, but it only raised me like 0.03, and I was like, what the F? And then when I did bad on the homework, it raised me down like 2%, and so I was like, you suck.
Those tests take forever too. Like that test still took me a really long time.
Mhm.
It didn't take quite as long as it took me last time, but it took me like a solid, I think, 4 hours. So it was long.
I think I like took for 3 hours or something like that.
So are
And
you
that was
guys
only because of time limit.
so you guys wanting to go over 4 or skip it and like go over it individually or what?
We should.
Honestly, I don't even know for.
I don't know. I don't know how to do this one either.
I thought I got 4, but I didn't get it right on the key, so I don't want to like. I know that they're supposed to match up like in a cross. Um,
And on the key, it almost looks like it's kind of doing the same thing as 3, but in a really weird way.
Mm-hmm.
Yeah.
It says it undergoes an alternate pattern of segregation in meiosis 1.
I know. Okay, so I know that it's supposed to. They're supposed to match up and make like a cross like that. I don't think my chromosomes are right, but it's supposed to make a cross. And then alternate is when they separate diagonally. So these two go together. And then adjacent one is right. Adjacent one is I think when they separate like so the two go together and those two go together. And then adjacent two is when the ones on top go together and the ones on bottom go together. But that's all I know. That's all I got. But you guys probably already knew that.
I'm gonna go through like all this stuff.
But do you guys understand how to like set up the cross?
Yeah. Once I know what I'm crossing.
I'm gonna have to watch the video first.
Is that on the video?
Mm-hmm.
Oh, good. I like that.
Yeah.
But for number 4, because there's like four of them, like which one do we cross with which one? Do we just cross the outside ones with the other outside one, and then the 2 that are in the middle with each other?
Well, that's what you're determining. So it makes like, at least I looked online, and the cross isn't like not like a Punnett square cross or anything like that, but like a.
Well, okay, these questions are only asking what the pattern is. So adjacent 1, adjacent 2, and the alternate. It's not asking you to actually cross them.
Yeah.
I'm pretty sure.
Because the key, it's asking for the chromosome order. So the product. So it's asking like. Where am I?
So on A, which ones are we crossing?
So that's what I'm trying to figure out. So like I know that in order to figure out which one is the alternate, like alternate is whenever the chromosomes diagonal to each other in the cross separate, and they're the ones that are together in cell 1, and the ones that are the opposite diagonal are in cell 2. But I don't know how to set up the cross to where I know which chromosomes go together.
Mhm.
So that's what it's asking. It's not asking like whether, because it's telling us this is translocation is undergoing an alternate pattern. So it's not asking us whether it's going through an alternate pattern. It's telling us that it's doing that. It's asking what product is going to be in each cell. But I don't know how to, like, I set up my cross wrong and I don't understand.
Oh.
Yeah.
So wait, so we're just crossing, so for A, we're crossing A, B, C, D, E, F, G, H, I with which one is this? With the one with the R, S, T, U, V, W, X, Y, Z, right?
Yeah.
I have no idea.
I'm so lost. Sorry guys, I'm not contributing much. I'm just so lost.
No, I'm really confused because I thought I understood this and I don't.
Yeah. I'm looking at the key right now and I'm like, I'm gonna fail this section.
No, we just gotta figure it out. So, because I looked up what a cross is, and I thought that whenever you make the cross, so this is on the video, like she actually makes a cross like this.
Okay, this makes sense to me, you guys. But I watched the video and I know how to cross them. But it's just the patterns. I just need to know what the patterns mean. So I need to go back through my notes and find that. But this makes sense why, like, it's just, yeah, all you're doing is you're picking, okay, so if it says alternate pattern, you're just picking the ones that are going to cross during an alternate pattern. You're crossing them, and then you're putting them down in the thing. You're just saying this cell's going to have this, and this cell's going to have this.
Does she do this? Is this what she does? Does she use a different method, or is this what she does for the whole cross thing?
No,
like,
no, it doesn't look like that. No. Just watch the video for sure. If I knew what like the differences, I need to go back there and find like what an alternate pattern is and all that stuff. But if I knew like what the differences were between those, I'd be able to figure this out. But I'm struggling because I don't know what the patterns are. But I know how to do the crosses. Like I know how to make them cross.
How do you do the cross?
Video. Seriously, it's like an 8-minute video on how to do them.
Okay.
Otherwise, it's gonna tell me like 8 minutes, or it's gonna take me like 8 minutes to explain. Like, actually, it'll probably take me longer because I don't know. But like, looking at these in the key. Oh no, that's not the key. It'll make a lot more sense when you watch the video, though. But if,
Let's just go to the next one then.
okay,
Yeah.
okay. So 5. Now I need to go back there and figure out the rest. But okay. So in this question, you'll examine non-disjunction. The first part of the question asks you to draw the results of non-disjunction in meiosis 1. The second part asks you to draw the results of non-disjunction in meiosis 2. Oh my gosh. In your drawings, diagram the behavior of 2 pairs of homologous chromosomes. Use color to distinguish the maternal and paternal chromosomes. In your drawings, also diagram the nuclear membrane, the microtubule spindle, and the metaphase plate.
And then she said, I will provide a drawing for this question.
What?
Yeah, and she put a note.
She put in a note that says, answer, I will provide a drawing for this question. I think I understood this pretty well, though. Did anybody else have any problems with it?
Well, we were supposed to, like, this is the one we're supposed to do together.
Oh.
Oh, are we still doing that rule?
Okay.
I thought she put a note to speak about that.
I didn't use colors, but whatever.
Hold on, let me open up my island thing. I could have sworn she put a note about it. Maybe that was from last week, but I thought it was from this week.
No,
I
it
thought
was
it
from last
was like
week,
a
but I
continuous
didn't
thing
know if
for
it
like
was.
from now on. We're supposed to do like number five together.
Oh, okay.
Oh well.
Well, so in metaphase, the homologous chromosomes line up on the plate.
Mm-hmm.
So that's metaphase 1. And then the non-disjunction would happen during anaphase, right?
When they're getting pulled apart.
Yeah.
Mm-hmm.
So it would like attach to every chromosome but one. So there would be like one extra in one cell,
On one, yeah.
right?
Mm-hmm.
Mm-hmm.
Okay.
And then telophase and cytokinesis is when they separate into the two cells.
Yeah.
And each cell would have like a different number, right?
Yeah.
A different number, what?
Of chromosomes.
Oh, yeah. So then in metaphase 2, each product would line up on the plate again. And then in anaphase, there wouldn't be non-disjunction, so it would pull all the sister chromatids apart, right? Like it wouldn't separate it weird again, right?
Mm,
Or the thing that I was confused about, like, because in metaphase 1, I put like, so there's metaphase 1, it separates all weird, and then so there's 3 right here, and then they line up again. So if that was the product, like if there were 3 chromosomes and those are the homologous, whenever they line up for metaphase 2, would they line up in a line like that, or would it line up like that and separate weird again?
I think
It
they
would
probably.
separate weird again because then there's another. It's kind of like the diagram on the PowerPoint slide because one of them wouldn't separate, right?
In metaphase 2,
Yeah, on the second part.
Yeah, it's on like slide 6 that I'm looking at right now.
so even if there's like a non-disjunction in meiosis 1,
Mm-hmm.
it affects.
Will affect it all the way to the end.
Yeah.
Okay.
That's why you come out with those diseases at the end, is because of those extra or missing chromosomes.
So it would end up like whenever it pulled it apart, those 2 homologous chromosomes. That's what I'm confused about though, because in metaphase 2, you're pulling apart the sister chromatids. So if those 2 homologous chromosomes are still connected to each other, then would it separate the homologous chromosomes and then one chromatid in anaphase 2? I need to look at the PowerPoint. I didn't look at the PowerPoint, so I'll just go look at that. It's okay.
I know I'm looking at that right now too. I'm trying to figure this out.
Mm-hmm.
Even with the PowerPoint, I still didn't understand because chromosomes is like my weakest point in everything of this, which sucks because the whole class is genetics. So
Centered around it.
Well, like I understand what happens in metaphase, like in meiosis 1 versus meiosis 2. I get that. But like if in meiosis 1 there were one pair of homologous chromosomes that didn't separate, and then there was one chromosome that did separate from its homologous pair. So there's 3 chromosomes.
Yeah, so what? Yeah. So, okay, I'm looking at the PowerPoint, and what you have is the 3, right? The 3
Yeah.
in one cell and the 1 in the other cell.
Yeah.
If it underwent non-disjunction in meiosis 1 and then normal, like, separation in meiosis 2, then yeah, those 3 in the one cell would all line up in a row, and you'd end up with 2 cells that have 3 like chromosomes, and then this cell would have 3 chromosomes, and then the one that had 1 in it that splits would have 1 and 1 chromosome.
Okay, that's what I got.
Yeah.
Sweet.
And then the other one, the meiosis 2, which goes normal meiosis 1, and you have like a normal, 2 cells with 2 splits in them or whatever. And then those are getting split apart. So the one gets split apart normally, and it has 2 and 2, and then the other one gets split apart weird, and it has 3
Like,
1.
okay, that makes sense. So, like, for the non-disjunction and meiosis 2, the product would be like there'd be 4 cells, and one of them would have 1 chromosome, one of them would have 3 chromosomes, and 2 of them would have 2, or I guess it's,
Yeah.
chromosomes.
Yeah.
And then I'm sure you could probably draw it out so that you would have non-disjunction both times, and then you get some weird answer depending on how it all went down. But like you could have non-disjunction, I'm sure, like in meiosis 1 and meiosis 2. I mean, I don't know how common that ever would be, but you'd get some like weird amounts.
Okay.
And like weird arrangements. So.
Well, that works for me. I need to get changed for work.
Okay. I need to take a nap. For
Like,
real.
I wish I could take a nap.
for real. Enjoy it while you can.
Oh my gosh, I will. While I can. Whatever.
Thanks, girls. You guys are such a help. I was so lost.
Me too.
Weirdly, this lesson makes probably the most sense to me out of anything that we've done so far.
I think I should watch the video since
Yeah, me
that's
too. I think that's like
Yeah.
Yeah. Okay.
the defining factor. Yeah. Okay. See you guys. I'll post the video.
okay.
Okay.
Bye.
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 Bye bye.